All engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
questions,
Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists, the programme that brings you the latest breakthroughs in science, technology and medicine with me, Chris Smith.
And with me, James Titko. Coming up, how scientists are growing new blood in the lab, the scientists planning for potential alien communications, and why fertiliser may be fooling bees' ability to spot flowers.
The Naked Scientists podcast is powered by ukfast.co.uk.
A new way to produce transfusible blood in the laboratory is being developed by UK researchers. Every day, a country the size of ours gets through about 2 tons of donated blood. At the moment, all of it needs to come out of someone else's arm.
But now, researchers have found a way to grow more blood cells from donated blood that could be used to top up and even maybe one day replace traditional supplies. Cedric Givert from Cambridge University's Department of Haematology is one of the team behind a trial to put these lab-grown blood cells through their paces in real patients. So how do you grow this new blood, Cedric?
What we have to do is to take stem cells that are present in anyone's blood, including donated blood, and we put them in special solutions with growth factor. It's a bit like growing plants and feeding your plants. And then over a period of 20 days, thereabout, they will multiply, but they will also change their nature from a stem cell to a mature red cell.
And do they, to all intents and purposes, look like, smell like, taste like and work like a normal red blood cell?
Absolutely. We put them through their paces in laboratories through a series of tests before the clinical trial to make sure they absolutely behave the same way as a donor-derived red blood cell.
And you're obviously sufficiently happy that this works well enough and safely enough, you're now going to put these into real patients?
We are actually starting very small with healthy volunteers, and we only produce 2 teaspoons of those red cells to then do this clinical trial that is currently ongoing. We want to check whether these laboratory-produced red cells last longer in a circulation compared to donor-derived red cells.
Why should they?
It's simply because donated blood contains red cells that are newly born, but also red cells that have been circulated for 2-3 months that are very old, and they will disappear from circulation quite promptly. Whereas the cells that we produce in the laboratory all are born at the same time, so they're all young, so they should survive in the circulation on average much longer than a donated blood.
These cells that we're talking about, though, they are fractions of a millimetre across. How on earth are you going to track a teaspoonful of those over time to work out how long they are there?
We label them with a very, very small amount of radioactivity before we re-inject them. And then we take serious blood sample from the volunteer taking part in a trial or receiving those blood cells. And then we use a machine to detect these very, very small amount of radioactivity. Over time, that radioactivity will fade because those red cells will gradually disappear from circulation. And as we measure the fade, that gives us an idea how long those cells survive in circulation.
And the advantage would be if you have cells that live longer, a patient who needs a blood donation wouldn't need their blood transfusion as frequently because what you're giving them would last longer.
Absolutely. This is absolutely transformational for patients who've got genetic disorders whose life really depend on being transfused regularly. At the moment, they are transfused every 2 to 3 weeks. We could potentially transfuse them every 6 to 8 weeks.
You're obviously comfortable that these cells are safe. One always worries when you take stem cells and then you grow them and stimulate them in the lab that they then turn into things that you would rather they didn't, like cancer cells, for example. What checks and balances are there to make sure that can't happen?
One of the big advantages that red cells don't contain a nucleus, so they can't multiply, they can't become cancerous.
But the stem cells can.
The stem cells can, but the purification of the product that we make in the lab at the last step takes away any cell that contains a nucleus.
So it's just the blood that's left over, so it should in theory be very safe. How much can you make? I know you're saying in this case that it's going to be a couple of teaspoonfuls for the purpose of experimentation, but if this is going to be practically used, you're going to have to make a lot of blood. How scalable is this?
It is scalable. Absolutely. And manufacturing large quantity of cells is something that is really taken off in the last decade. But in particular, the pandemic has shown us how much more we can scale cell cultures. In this case, it was to produce the vaccine. But some of those technologies are directly applicable to what we want to do with the red blood cells.
Well, we wish you luck, and you can come back and tell us how the trial is going pretty soon. Cedric Gavert there from the University of Cambridge. Thank you.
Fertilisers are critical to modern agriculture. Without the higher crop yields they provide, we'd all starve. But fertilisers are typically made using ammonia, meaning they have a significant environmental footprint in the form of greenhouse gas emissions. They also produce algal blooms in rivers, reservoirs, and even the sea.
But now a new study suggests that by affecting the electric fields around flowers, fertilisers can also alter the way that pollinating insects like bees see them. Bristol University's Sam England told Will Tingle how plants generate their electric fields and what fertilisers might do to put bees off.
The electric fields around flowers kind of have two main sources that we're aware of. One comes from their interaction with the atmosphere. So these flowers are conductors to some extent, in the same way that the metal in your wires and your electrical appliances at home are conductors. Actually, plant tissue is quite conductive too. Now, the interesting thing is that these plants are obviously stuck in the ground, but they're pointing up above the ground into the atmosphere. And the atmosphere actually has an electric field in it too. And when this gets really strong, that's when you see things like thunder and lightning. But even in fair weather, there is still some amount of vertical electric field in the atmosphere. What this means is that the grounded plant, relative to the air around it, actually has an electric field coming off of it because it's not at the same electrical charge as the surrounding air. But also, in addition to these kind of electric fields that are generated by their interaction with the atmosphere, the plant itself also has all kinds of physiological processes going on inside it, where it's, transporting ions and signaling within the plant between cells that also generate an electric field external to the flower. So those are the two main sources of kind of electric field around flowers that we know of at the moment.
And how would insects such as bees use these electrical fields to detect the presence of flowers?
Something that's really cool and that we've only really discovered over the last decade or so is that a couple of different species of insect are able to detect electric fields in air. And the way that they do this is that basically hairs on their body get pushed around by the electric field. If you think about the same way as when you charge a balloon up with your hair, the balloon can kind of push your hair around. Essentially, the same thing is happening with the tiny little hairs on these bees as they're entering into the electric field around flowers. And the cool thing is that it might actually provide some useful information to them. One, in theory, they could be using it to just detect the location of flowers, but of course they have excellent senses of smell and also vision that help them do that. But something that we have discovered previously is that when a bee visits a flower, it modifies that electric field around the flower for some amount of time, on the order of a few minutes. And so then a bee that comes along after that first bee might be able to detect that difference in the electric field and essentially use it as a cue as to whether a bee has visited that flower recently, and that maybe the nectar might be diminished because it's been visited recently. So it might basically give these bees a bit of a clue as to whether there's a good amount of nectar in that flower or not.
What is it about synthetic fertilisers that alters the detectability of these flowers?
We try to unravel this a little bit in our study, but for sure this is something where the exact mechanisms kind of need to be delved into in a little bit more detail. We think there's probably, again, two main ways that this is happening. One is that it just modifies the electrical properties around the surface of the flower, but also in the air around it by changing things like the humidity, but also the conductivity, like the electrical conductivity on the surface of the flower. And this essentially means that the electric fields kind of travel in a different way through the material, which can make them stronger or weaker. And in the case of the fertilizers that we looked at, it seemed as if they actually make the electric field stronger, which might be quite confusing to the bees. The other way that it might be having an impact is that a lot of these harsh chemicals that are found in synthetic fertilizers can stress the plant out. And when they do this, that will basically cause the plant to start moving ions around, kind of signaling within the plant between different cells to essentially tell other parts of the plant that, oh, maybe we're in a bit of trouble here, which again, that will change the electrical profile of the flower.
Finally, what would be the ramifications on insect populations and their relationship with flowers because of this discovery?
This is one of those things where there's really a huge arsenal of evidence that's beginning to accumulate, that there are just so many different ways that a lot of our activity and a lot of the chemicals that we're introducing into the environment are having quite adverse effects on quite a few different species of insects. We already know that a lot of fertilizers and pesticides are quite harmful towards pollinating insects like bees. And our study really just adds another dimension to how that's happening. But of course, that does somewhat optimistically open potentially new avenues for how we can try and mitigate these negative impacts in the future as we continue to develop these technologies.
Really interesting, isn't it? One of the most fascinating studies I've heard about in a long time. That was Sam England. He published that work in the journal PNAS Nexus. Humans have always looked up at the stars and they've wondered whether we might be sharing this universe with other forms of intelligent life. Well, one of those wondering is St Andrews University's John Elliott, who coordinates the newly launched SETI Post-Detection Hub. That's the Search for Extraterrestrial Intelligence. And he told James why we need to have a plan in place for when the inevitable close encounter he's envisaging occurs in the future.
The Post-Detection Hub is when we've discovered something, whether it's a distant signal or even an artifact, a myriad of scenarios, but it's the bit where it says what next. And we're the what next. We need to coordinate all the different disciplines, all the different sort of scientific endeavors together, such as we have a framework, a methodology by which we then know whatever scenario it is, we have a plan how to deal with it.
I'm sure, John, you get bombarded with movie references. But what springs to mind when you talk about this coordinated effort is Close Encounters of the Third Kind, when they're receiving those light and audio signals and the team of government-led officials are there furiously typing away, trying to crack what the communications are.
The whole point of the plan is that we'd have forethought in what we need to do, and we'd have done many years of research to plan this. The scenarios you mention in films often are, let's get the code breakers involved. No. An analogy is in the Second World War with the Enigma code, where we were cracking a German code. Now, we knew it was German. All right, it was encoded, but we knew what the source language is, and we knew we had to try and translate it into something we understood. We're dealing with the unknown, and everything I've created, mathematical models, algorithms, all the dirty stuff that's underneath it all, has to deal with how can we make sense of something we know nothing about in the first place.
Probably difficult to get my head around just the basic principles you have to start from. But could you give me an idea? Obviously, it's going to depend on what sort of communication we get for you to then be able to do something with it. What sort of eventualities have you prepared for so far, for example?
If we receive something, just say like a binary bit stream, loads of ones and zeros, if you looked at it, billions of these coming in, no one could make sense of it. And if it had no resemblance to any human language as we particularly know it, you couldn't translate it into something. It wasn't like making up your own Rosetta Stone. I've devised algorithms that slide along it with variable windows. It's pattern finding. If there is information type structure in there which would flag up intelligence, you already know that you've got something special, and you build up and you try and piece it together like a jigsaw as best you can.
It feels like, as a species, people are coming round much more to the idea, as the scale of the universe becomes more apparent to the layman when they see images from the James Webb Telescope, better science communication from astronomers. There's almost a building consensus that intelligent life is a firm possibility.
What's happened more recently is we've just been discovering more and more these planets around distant stars. So what we call exoplanets, we're finding thousands of them. In fact, we're finding them really around every other star. And if you think about just our own galaxy having, say, at least 200 billion stars in it, we're talking then the same sort of numbers for planets out there. In fact, if you had multiple planets around most systems, you may be going towards 1 trillion planets in our own galaxy. That's just our galaxy. There's 125 billion in the observable universe of those. To think there's no life out there, I think there's even life probably microbial in our own solar system, but intelligent life, within 1,000 light years of where we are, we've probably got at least a handful, if not more, potential intelligent civilizations sitting out there.
It's all thought-provoking stuff, isn't it? John Elliott there, and you can keep tabs on their mission via their website, which is at fromafar.world. Yes, slightly unusual address, fromafar.world.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to The Naked Scientists with me, James Titko, and Chris Smith. Still to come, the sharks doing reconnaissance for marine biologists. But before that...
Good news, because this week scientists at the University of Cambridge have found some markers in the bloodstream that can flag up individuals on the verge of developing diabetes. Intervening early in cases like this can help patients to take action sooner to avoid the consequences or not even develop diabetes at all. But existing traditional techniques for doing this sort of test can be very unreliable. They're also inefficient. The new work can help to change all that. And with us is Julia Carrasco Zanini, who did the work. So you better tell us what the test involves.
So there are a subset of people with prediabetes that can only be identified through a very inconvenient and time-consuming test. So this is called the oral glucose tolerance test, which essentially involves giving people a sugar drink and then 2 hours afterward taking a blood sample and measuring their blood glucose levels. Now, the new test that we're proposing would only require a one-off blood sample to measure in there 3 proteins that would enable to predict the outcome of this oral glucose tolerance test.
So these are chemicals that are washing around in these people's bloodstreams, and you're saying if we measure those, they can predict what would happen if we were to feed those people a whole heap of sugar and tell us if they are at risk of diabetes.
Yes, essentially, like those 3 proteins would then enable us to classify individuals that would be predicted at high risk of having this specific type of prediabetes.
How did you find the 3 chemicals? Because there are obviously millions of different molecules in the body. How did you home in on those 3?
So we have a really large study with over 11,000 people in which we performed this oral glucose tolerance test, but in whom we'd also taken a prior blood sample where we measured around 5,000 proteins circulating in blood. So then the question really was, out of these 5,000 proteins, can we identify those markers that are very informative to do this prediction? Obviously, every single time that we want to make the prediction, we don't want to measure again 5,000 proteins. So then for this, we developed a machine learning framework, which essentially enabled us to extract those 3 proteins that were considered the most predictive of the outcome of this oral glucose tolerance test.
I suppose you're sort of sorting through a biochemical haystack here, aren't you? You're saying we've got millions of molecules we could consider. You homed in on 5,000 to start with, and you're just saying, what of those, when the person has this situation where they're at risk of developing diabetes, which ones consistently are changed, either up or down, relative to a normal person? And those are the ones you then have found that are the most predictive.
Yes.
And how reliable is it?
Well, so this has a very good impact since we've estimated that measuring these 3 proteins, if we were to implement it along with diabetes screening strategies, such as the NHS Diabetes Prevention Programme, we could actually reduce the number of oral glucose tolerance tests that would need to be performed to identify one of these people with prediabetes by half.
Oh, it's quite a big difference. How long before a person develops diabetes would they go positive on your test?
So this is one of the things that would require further follow-up studies and trials that are large enough specifically developed to assess the effectiveness of these 3 proteins.
Because there are lots of databases available now, aren't there, where people have, I mean, the UK Biobank is one where over the last 20 years, people have given blood samples, had very close follow-up, and we've got good histories on what happened to those people. Presumably you could go to big data sets like that and now begin to comb through those, looking at the same molecules and test it that way.
Absolutely. I think it's really important to consider absolute risk in people that have high levels of these three proteins, yes.
So what sort of a difference could this make then? If this did become policy and they did decide to turn this into a test, we can look at the practicalities of that in a moment, but what sort of difference could you make if this were implemented instead of the current practice?
Well, so essentially, it would enable us to identify in early stages of the disease people who are at very high risk. And then, of course, this would be very beneficial because it is known that behavioral and lifestyle interventions are really effective in delaying and preventing the onset of type 2 diabetes.
Oh, so you could give people preventative advice. So if I did this test and it found that I was at higher risk of developing diabetes, you're saying that would give me advance notice and I could make changes that would reduce the chance of that happening?
Yes, absolutely. And it's currently something that, because these people remain undetected so long, they don't really know that they are at high risk and that they should do these behavioral modifications.
So we're sort of in those people slamming the door after the horse has bolted.
Yes.
Whereas with what you're doing, you give people some warning beforehand, so there's still time to intervene meaningfully.
Yes, absolutely.
Is it easy to detect these proteins? Because at the moment you've got a powerful laboratory to do this. If this were to be turned into a test, how practical is that?
So this is one consideration that has to be taken into account before thinking this could be implemented in clinical practice, because how we measure these proteins in blood actually really matters. So in our study, for example, we compared the measurements for these three proteins with two different technologies, and we found good agreement between them. However, if we were to roll out this more widely, it is very likely that we would have to develop a single, robust, universal test that could be rolled out in a wide range of clinical settings.
Does it miss any cases? Because that's the other important thing, isn't it? It's very good to have a test that finds people really fast and gives us loads of lead time. But if it misses critical cases, that's dangerous.
Absolutely. I think this is one of the things that would really require a larger trial to be able to specifically say how effective are these three proteins and how many people they miss and how many people they identify, and ultimately the cost effectiveness of a strategy such as this for healthcare systems.
Giulia, thank you very much for telling us about your research. That's Giulia Carrasco-Zanini. She's just published the work she was describing there in the journal Nature Medicine. The field of marine biology has been recruiting new members to study and measure marine life. And those new members are the marine life itself. A study off the coast of the Bahamas has discovered the largest seagrass colony in the world, so large in fact that it pushes up the amount of known seagrass on the planet by 40%. This seagrass was discovered only when a team of scientists strapped cameras to tiger sharks, and that's no mean feat, believe me, and watched where and how they went about their day. Ollie Shipley from the organisation Beneath the Waves spoke to Will Tingle about their new recruits' findings, including the obvious question, how do you tie a camera to a shark?
The way that we explain it to people, it's almost like a NASCAR pit crew. And what we basically do is we strap the camera or the package, because it's not just a camera, remember, we've got other sensors. We've got kind of a satellite tag, a radio tag on this package as well, so we can find it when it falls off the animal. And we use biodegradable cable ties to basically affix that camera package through the fin of the animal, and the cable ties are secured by a piece of equipment that's called a galvanic time release. And what that is it's basically a dissolvable link that dissolves in seawater, and the kind of dissolution time is pre-programmed. So that thing will dissolve within, about 4 to 6 hours typically. And then the whole package basically falls off the animal. And through a satellite and a radio tag, we can basically then go and locate that package once it's off the shark. We then remove all of the hook, take the cables off, and let the animal kind of swim on its merry way. So all of that happens, in about 8 to 10 minutes typically.
Given that all of the technology we have, all the sensory equipment we have available to us, satellites and ocean scanning equipment, why did you feel the need to strap a camera to a shark?
We've been putting tags on animals for the last 30 years, 40 years, some of these satellite tags. But we really don't know a lot about what goes into their daily routine. And I think the way that the technology has developed over the last 5 to 6 years, we're now able to explore and observe high precision information on what these animals are doing every minute, of every day. For this study, whenever you strap a tag onto an animal, it's likely going to take you to a really important place, because these animals are making calculated decisions every day of their life. They're trading off energy, versus the cost of actually moving from one place to another. And so all of their decisions are calculated. So the chances are, if you strap a tag onto an animal, it's going to take you to somewhere that's pretty important for that individual or that population or that species. For this study, we kind of utilize that concept, and we used the cameras strapped onto these tiger sharks or attached to tiger sharks to basically quantify the density of seagrass that they were swimming over. And what we were able to do from that, we were able to extrapolate those kind of seagrass density measurements and use it to ground truth information from satellites to basically quantify or measure the largest seagrass ecosystem in the world.
And when you say ground truthing, what does that mean?
Ground truthing is basically confirming, because a lot of these measurements are made from satellite, and there's sometimes quite a bit of error associated with that, and that can be due to things like cloud cover, or if the water isn't particularly clear on a certain day, for example. And so you actually have to go in there and empirically measure the density of the seagrass in this case, so you can confirm that what the satellite-derived data is telling you is true.
What have the cameras on these sharks revealed to us about seagrass distribution?
Well, they've revealed that the area that we were working in, the Bahamas, houses an incredible abundance of seagrass. We had an idea that it was there, but we weren't really sure how much. And so through strapping these cameras on these tiger sharks, they've really revealed how kind of expansive and dense these seagrass ecosystems are on the Bahamas banks. And it's only through that we're able to confirm, that this is the most significant, largest seagrass ecosystem in the world, which is crazy.
And do you know of any other shark camera equipment that's going on? Maybe new discoveries could be made somewhere else.
This is a technique that has been utilized in a few different species before. So there's a group that are using them in sea turtles to kind of map similar things, because sea turtles also interact quite abundantly with seagrass meadows. But I think this is where the kind of innovation really gets exciting, because I think we have, this is a proof of concept that has led us to an incredible discovery. And so, I think the world really is our oyster in applying this sort of technology to other animals as well. And we certainly have plans to do that in various parts of the world.
Ollie Shipley, and that paper can be found in Nature Communications.
And that's all we have time for this time on The Naked Scientists. Join us next time when we'll be looking at reproducibility in science. Why is it that 70% of scientists claim to not be able to reproduce another scientist's results? Join us then to find out. And after that, we've got a Q&A in store, so we're giving you plenty of time to get those questions in. Chris will be joined by an astronomer, an archaeologist, a physicist, and a global health expert. So do be sure to send your scientific queries to chris@thenakedscientists.com. That's chris@thenakedscientists.com. The Naked Scientists comes to you from the University of Cambridge's Institute of Continuing Education. It's supported by Rolls-Royce. I'm James Titko. Thanks for listening, and until next time, goodbye.